Hi everybody, and welcome to VIBS 243, and welcome to the first TA review. So this is me, Jubilee Brown. I'm one of your many TAs, and I will be doing the TA reviews every week, recording them and posting them on eCampus. So I'm glad that you made it here and are able to watch this. Since this is the first week, I will kind of go over what these TA reviews are going to be like every week. So this week in class, the first week that we're actually covering content, we are going to be going over Introduction to Microscopy. And so there are two different sets of notes for that, and two different lectures from Dr. Johnson that go with that. And then if you did check, I did also do a kind of TA version of the Intro to Microscopy 1. And that is just because I'll let you guys know Dr. Johnson does cover a lot in his lectures that you will not be tested on yet. Okay, so I know a lot of you guys get stressed out and stuff because he starts labeling what these cells are. He starts telling you everything that's going on in these cells. And it can be very stressful because you think that you need to know all of that yet. So I will go ahead and just tell you for this quiz, you do not need to know all the names of these cells and everything like that yet, or what they do. Those will be things that we will cover later in the semester, that you'll be responsible for later in the semester. So for this week, the main things that we're gonna want you guys to know are gonna be kind of the things that I covered in the TA version of the lecture for Intro to Microscopy 1. Whatever Dr. Johnson covers in Intro to Microscopy 2. And then I'm kind of gonna be hitting the main points, but more of a summary version of it in this TA review. Okay? So hopefully that makes sense, but basically what I'm saying is just don't get stressed out when he starts pointing out all of these cells, all the different white blood cells, all the different organs and everything and what all they do. Right now we're mainly focused on introducing you to microscopy, so understanding how cells and tissues are stained, how we can observe them, and stuff like that is going to be our main focus for this week. Okay, so just kind of getting you used to what we're going to be looking at for the rest of the semester, and how we can actually study tissues and cells on a cellular level. Okay, because that is the basis of this course. That's how we do histology. Okay, so yeah, and I'm going to be doing the reviews every week. I will post them, hopefully towards at least by Monday, hopefully Monday by end of day every week, for you to watch them. You can go ahead and watch them before or after lecture, whatever you prefer. And then I will post a short TA review assignment to go along with that. And that's just to make sure that you did watch the lecture. And so you can do that really quickly, turn it in, and that will get you those 5 points of extra credit for watching this review every week. Okay, so we'll go ahead and get started with the review. So again, this week what we're learning is just Introduction to Microscopy. Because obviously, so this course is about histology, right? So it's about studying tissues, organs, organisms, organ systems, but on a cellular level, right? On a cellular level. And so in order to do that, we need to view these things through micrology. So this is just kind of a review from biology. Hopefully you guys have seen this before or heard of this, but this is just kind of how we classify different tissue levels and stuff like that. So if we go from the most, or I guess the smallest kind of term for anything that's living, that would be protoplasm. So protoplasm is the term that we use for anything that is a living substance, okay? And so that is just kind of a definition thing that we do need you guys to know. It's super straightforward and stuff, but people do tend to forget it just because it is so straightforward. knowing these kind of definitions. So protoplasm is just any kind of living substance. The smallest unit of living substance or protoplasm that is able to maintain life by itself. Okay, so without any other units of protoplasm or anything, it can maintain life by itself. That is going to be what we define as a cell. Okay, so a cell should be able to maintain life by itself independently of other cells, as long as it has everything that it does need in its environment. Okay, so that's cell, that's the smallest unit of protoplasm. Once you get a bunch of cells together, if a bunch of cells are together and they're working together, they're helping each other out with the same kind of function in mind and the same kind of texture, that is what we're going to call a tissue. Okay, so if you get enough protoplasm together, you get a cell. If you get enough cells together of the same function, you'll get a tissue. So that's going to be something like muscle, nerve, epithelium, and and then if you get enough tissues together, right, so if you get a bunch of muscle, nerve, epithelium, and everything together, you can create an organ. Okay, so an organ is two or more types of tissues that are working together. The tissues all have the same kind of goal, and then they're all going to work together for a larger functional goal. And that's what's going to make an organ. So that's something like the kidney, the brain, the liver. They all have the same big functional goal, and they're all made up of at least 2, but more often 4 types of tissues. Okay, and we'll go over the different tissue types in a second. Once you get multiple organs together, right, and they're all working with the same larger functional goal, that's where you get an organ system. Right, so your digestive system, your skin all has the same functional goal. Your endocrine system all has the same big functional goal. So that's when you get multiple organs together, you create an organ system. Once you get multiple organ systems together, they're all working for the greater good of an organism. Right, so you have multiple organ systems, that creates one entire organism, like you, me, your cat. And so these are kind of just the levels of organization. It does sound really straightforward. Just make sure that you do understand the definitions, right? So you get protoplasm, enough, you get a cell. You put enough of those together, you get tissues. Then you get organs, organ systems, organism. Okay, so that's kind of the hierarchy of these different tissue types. Okay, so we're going to be focusing we'll be focusing on all of it. So we'll be focusing on the individual cells, what they do, how they help the tissue, how the tissue helps the organ helps the organ system, and how that helps you in general stay alive. Okay. So this right here, I will just let you guys know. So Dr. Johnson goes over in great detail, all the different types of white blood cells and everything like that. And so I will just let you know, we will not be testing you on the different types of white blood cells, okay? We will cover that later in this course for sure, and at that point I will expect you to know the difference between all of these white blood cells here. But as of right now, I do not need you to know the difference between a neutrophil, an eosinophil, a basophil, a lymphocyte, a monocyte, okay? Just look at these and just kind of understand how we are able to study these through microscopy, right? So it is through microscopy I'm able to actually look at these white blood cells and be able to differentiate between a neutrophil and an eosinophil, okay? You don't have to know how to do that yet, but it just goes to show you that if you do have the knowledge, microscopy is what's going to help you differentiate these two, because obviously I can't see white blood cells with my naked eye. I need microscopy to do that. So this is going to be light microscopy. We'll go over it in a second. And then this is going to be electron microscopy of the same cells. Okay, so it just goes to show you, we can visualize all these different cells. We can visualize them in different ways. And the way that we visualize them will give us information about all of these cells. So we can tell what's going on. We can tell what these cells are doing, what they look like, how they function in the overall bigger picture of an organism. Okay. So when we think about microscopy, there are two big things that we think about in terms of being able to visualize and picture cells, and being able to picture things that are very small, and that's going to be magnification and resolution. Okay, and so these are just the definitions, and I do need you guys to know that. So magnification is simply going to be an increase in image size. Okay, so it literally is just making things bigger. Okay, so this picture behind here, this is actually the lining of the stomach. Okay, and if I literally just take a chunk out of here and I just make it bigger and bigger and bigger, and we zoom in, it's going to look something like this. Okay, it's just going to get bigger and bigger and bigger, but it's also going to get more and more blurry. Okay, because I can't differentiate, between this little smudge and this little smudge, between this white little smudge and this white little smudge. In order to be able to tell what's going on here and to differentiate between all of these little smudges, what we need is a higher resolution. Okay, so magnification is just making it bigger, not necessarily any more clear, but just bigger. Resolution is what's going to make it more clear. Okay, so resolution is the smallest distance between 2 points that can be distinguished, right? So if I wanted to increase the resolution, I could do so, and then I would be able to tell, what's going on with this smudge versus this smudge. Right here, the resolution is not very great, so it all kind of looks the same. There's not very much differentiation or clarity going on in here. So in order to be able to visualize these cells and everything that's going on in here, I would need to increase the resolution. Okay, so these are two things that we're obviously concerned about when it comes to microscopy. Is yes, we want a higher magnification so we can see these cells, but it doesn't help unless we can get a higher resolution as well. Okay, and so one of the first ways that we do visualize cells is going to be through light microscopy. That is where light is used as the source to bounce around these tissues and be able to differentiate between this cell and this cell. Okay, so it employs visible light, and we're using that to increase the magnification, increase the resolution to be able to see these tissues. But in order to do that, we have to cut these tissues very thin, and when they are very thin like that and we're zooming in on them, they are actually transparent. Okay, so this here is actually just a sample of tissue that was stained improperly, so they didn't stain all of it. So this part actually didn't really pick up the stain very well. This part obviously did. So that just goes to show you, if we do not stain the tissues that we're trying to look at under the microscope, they're not going to show up. We're not going to be able to see what's going on. And you can see here how once we've stained this, we can see the overall structure of what's going on here, and how these cells are working together to create a tissue or an organ. Okay. So when we think about light microscopy stains, there are three main stains that we want you guys to definitely know. These are the most common that we use in histology in order to study tissues. Okay, so these are the three stains that I want you guys to know. H&E is going to be the first one. It is by far the most common and the most widely used one. So H&E is the first stain, and that's going to be this pinkish one here. It's very pink and purple. And what that stands for is hematoxylin and eosin. Okay, and so how that works is this hematoxylin is going to stain nucleic acids blue. Okay, so you have nucleic acids in your nuclei. You have nucleic acids in your ribosomes. So places with lots of nucleic acid material are going to stain a little more blue. So in here you can see this is the nuclei of a cell, and it is much more purpley blue, right? It picked up a lot of that blue. Here's another nuclei. It's a lot more purpley blue. Okay, so that's the hematoxylin staining those parts of the cells. And then eosin is going to be the other part of the stain, and that is going to stain our proteins red. Okay, our proteins red. And so you can see here, so we're looking at a tissue, okay? And so you can see here is a cell. Okay, don't worry about the name of the cell, but this is a parietal cell. And so you can kind of see the borders of the cell, and you can see this is going to be the cytoplasm of the cell, and it's very pink, right? So it picked up a lot of that red, a lot of that eosin, and that lets me know, based on the properties of this stain, that this is a very protein-rich portion of the cell, right? And that makes sense. We have a lot of protein in the cytoplasm, okay? Because that's what that cell is doing. It's making things. It has organelles in there, so it's very protein rich, okay? And then when I look at this more darkly stained circle in there, okay, that tells me that there are a lot of nucleic acids in there, okay? And that makes sense because this is the nucleus. So this is depicting the nucleus, and obviously it has a very high concentration of nucleic acids in there. So it's going to stain more purple, more blue. Okay, and so it is because these different compounds that stain different portions of a cell that allow us to see, you know, everything that we see, that allow us to differentiate this cell from this cell, this nucleus from the cytoplasm of the cell, okay, and so on. So that's H&E, that's the most common one, and it is normally going to look very pink and purple dots are what you guys are going to see a lot of. The next stain is going to be toluidine blue, okay, and hopefully you can guess which one of these is going to be toluidine blue. It's going to be this one, it's the blue one, okay, and so toluidine blue is what, so what it's going to do is it's going to stain tissues, and obviously everything is going to be blue, so there's no difference in colors, right? The H&E has two different compounds that stain two different colors. Toluidine blue, everything is going to stain blue, but they're going to stain in varying shades and varying intensities based on the density of whatever's going on in the cell or the tissue, okay? So you can see here, everything's blue, right, but we have some very dark portions, right, with higher densities in there, and then we have some very light portions and circles within this stain, okay? So that's what we look at with toluidine blue, okay? It's just the varying shades, the varying degree of color pigment that was picked up in different parts of the tissue, and that's what's going to help us kind of see what we're looking at, okay? And then the third one is going to be periodic acid Schiff. Schiff is the scientist or whatever that came up with this stain, so periodic acid Schiff. And so what this stain does is it stains carbohydrates or sugars, okay? So carbohydrates, it's the same thing as sugar, so anything that is very sugar rich in this tissue is going to stain this very dark, bright magenta-y color, okay? So in this one, you can see here, so these bluish areas are going to be the nuclei of the cell, and you can see this kind of outline around these cells, right? This kind of outline around these cells, and that tells you this is a very sugar-rich area, okay? Very sugar-rich areas here, and it's actually, so what's very sugar-rich and picking up the stain here is actually going to be the cell membrane. The cell membrane of these cells, the brush border of these cells and stuff like that are going to be very carbohydrate rich, and so it does give you a much clearer indication of the cell membrane, the border of these cells, and the distinction between those, whereas in an H&E stain that might not be as obvious, okay? The cell membrane and beginning of and end of each cell, it doesn't stain very darkly, because again, we're just staining nucleic acids and protein. Here we are staining carbohydrates, we're staining sugars, okay? So hopefully that shows you a little bit about what we're dealing with light microscopy. It just goes to show you that what we're gonna see in tissues is gonna depend on how we're staining it. Obviously what we're looking at is going to affect it, but how we're staining it matters. What stain we're using and what that stain is used for, so what that stain actually deposits its color for, okay? So it helps you to know that the more red things in an H&E stain are going to be the more protein rich areas, okay? So that's light microscopy. So these stains, what they do is they give you a lot of insight on kind of the structure, the overall way that these cells fit together and create a tissue, right? But what I would ask you guys is, do they give you a lot of cellular detail within a cell, right? So you can see all this pink fuzziness, you can see this purple nuclei, but does that tell you a lot about what is actually going on inside the cell as far as the organelles go? Does that give you a lot of cellular detail? And so what I would say is no. So in order to see cellular detail, light microscopy is good for overall structures and stuff like that, but if we want to actually see what's going on within the cell, if we want to see the organelles, the composition of the cytoplasm of the cell, we really do need to zoom in further. And to be able to zoom in further to increase that magnification, we are going to need to drastically increase the resolution, the clarity at which we can see things. And so in order to do that, we do need EMs. We need electron microscopy. Okay? So in light microscopy, we're using light as the kind of source to differentiate between this point and this point, this point and this point, this point and this point, right? But while light waves are small and everything, they are not as small as electrons, right? The frequency of electrons is a lot higher than the frequency of light. So electrons will actually give us a higher resolution because they have a higher frequency, and so they can actually differentiate between much smaller points, giving us a much higher resolution, if that makes sense, okay? So this is electron microscopy. So this is more used for cellular detail, for zooming in. So I do want you guys to be aware of the kind of level of magnification that we're at right now. So a lot of people will look at something like this and they'll be like, okay, is this a nucleus, right? Is this a nucleus? And they're trying to find a cell in here. So what I'm going to tell you straight off is you need to know the level of magnification that you're at, right? So these are not individual cells right here. We are looking at maybe a quarter or a fifth of a cell, of one cell. So this whole thing, this is all the cytoplasm of one cell. Okay, this line right here is the dividing cell membrane between three different cells. So this is a junction between three different cells. This is where they're meeting. Okay, so you just need to know the level of magnification we're at. We're at a very high magnification in these EMs. Okay, we're very zoomed in. So here you can see the Golgi apparatus. You can see little clusters of ribosomes. You can see vesicles, everything like that. You can see individual mitochondria, right? We would never be able to see that in light microscopy. Okay. So just make sure you're aware at the kind of level of magnification that we're at. And yes, so the organelle details are much more prevalent, right? We can actually see what all is going on in this one individual cell. Okay. So just like there are different types of light microscopy, there are different types of electron microscopy. And so all of these are just different ways that we can visualize these cells, that we can analyze them, see what's going on. Okay, so again, just like we had three big ones we wanted you to know for light microscopy, there are three big ones we want you to know for electron microscopy. These are them, and so the first one is going to be conventional transmission electron microscopy or traditional transmission electron microscopy, and that's just going to be the normal one, is how I tell people that. So all three of these are conventional transmission electron microscopy, and so it is very 2D. It's very flat. It's it's not 3D or anything. There aren't very many textures. It's just like if I took a little picture of it or whatever, and I saw through the cell, flattened it out and everything. So you can see everything. You have a whole bunch of cellular detail and everything, right? But it's not 3D. It's not like jutting out at you like our other electron microscopies will be, okay? And I will tell you, this one is the most common. So conventional, traditional electron microscopy, it is going to be the most common. It's the one that you're going to see the most as far as electron microscopy goes. but we do want you to know the other two. So the next one is going to be scanning electron microscopy. So both of these are examples of scanning electron microscopy, and so you can see how this is different, right, than the traditional. So this actually looks 3D, right? There's shadows and dimension to this, right? It looks like it's popping out at you, and you can kind of see the texture of these tissues, right, and everything that's going on. Okay, so it looks more like I scanned in a picture, like I actually took a picture of what's going on, and I can see three-dimensionally what's going on. That's going to be our scanning. Our next one is going to be our carbon replica transmission electron microscopy, and so it's just like it sounds. So carbon replica, the way that we're getting these is it's like whenever you make a cast of something, right, or you make a Play-Doh mold. So that's when, if you're trying to make a mold or a cast of something, you have whatever you want to make the cast of. You have Play-Doh or concrete or whatever, and you literally put that on whatever you're trying to make the cast of, right? So like if you're trying to make a cast of Bigfoot's footprint, you'll just put a bunch of concrete down there. You'll pull up the concrete, and you'll actually look at the concrete to see the shape of everything you're looking at, right? So that's what we're doing here. It's like we're laying Play-Doh down on the surface of these cells. We're peeling it back, and then we're looking at that Play-Doh. Okay, we're looking at that carbon replica and seeing what we see. So you can see once we do that, it gives you a lot of surface details, right? So you can see this is the surface of the cell membrane, the plasma membrane of a lymphocyte. And you can see part of it is peeled back, and you can see the cytoplasm of that lymphocyte. This is going to be the nucleus of that lymphocyte, so you can see the surface of the nucleus, the little nuclear pores that are in there. So it just gives you a lot of surface detail, okay? Like I can't see through the cell or anything like that, but it gives you a lot of surface detail. So it's literally like we're making a Play-Doh mold of these cells. We're looking at the Play-Doh, and we're analyzing whatever we see, okay? So those are our three types. So now we're going to practice, and hopefully you guys can identify really easily which EMs we're looking at, right? So if you look here, so which one is going to be the conventional one? Conventional transmission electron microscopy. Again, that's going to be the very normal looking one, right? The very flat 2D, two-dimensional one. And so that's going to be this one right here. When we look for scanning, that's the one that's going to be very 3D. It's going to give us a lot of textures, surface detail. There's going to be shading and stuff. It's going to be like we literally scanned in an image of what was going on. So that's going to be this one, okay? And then carbon replica is going to be this one, right? So it looks like a Play-Doh mold, a carbon replica of whatever we made. Okay. And so now we're going to talk about the 4 different basic tissue types, right? So we said whenever we broke down everything, right, you have cells. Those cells make tissues, and these tissues are going to make organs, organ systems, and organisms, right? And so with very few exceptions, pretty much everything in your body, in your friend's body, in your cat's body is made up of these 4 basic tissue types. They're all just combined and mix-matched in various different ways to make different organs, organ systems, organisms. Okay, so these are kind of like the big building blocks of what makes an organism. Okay, so it's going to be epithelium, nerve, connective tissue, and muscle. And we'll learn more about these in later units. Later we'll have an entire week on muscle, connective tissue, nerve, and epithelium. But we do want to know these basic tissue types. So epithelium is going to be this one right here. So this is epithelium. Epithelium is everywhere, and I'll go over that in a second, but it's basically just a lining. It lines things. It's like a transition piece. Nerve, so this is going to be a picture of nerves. So we've got a bunch of axons in here. We've got a bunch of neuron cell bodies in here. And then we've got connective tissue, so this is connective tissue. Fat is connective tissue, so this is kind of what glues everything together in your body so that you can't, just peel your skin off and fall apart. And then this is going to be muscle, so this is actually cardiac muscle. the muscle in your heart, and that's what's going to be in charge of contractions, right? So these are our 4 main tissue types, and if you combine all of these in various forms, you'll basically be able to make a person. You'll be able to make an organism. Okay. So the first one, so epithelium. So the definition of epithelium, it does confuse a lot of people, and so that is why I really want you guys to understand what epithelium is. It's not a specific type of cell or anything, it's a type of tissue, right? And so this tissue is found all throughout your body. And so what epithelium is it's basically a lining, okay? It lines things, it lines organs, it lines your skin, it lines blood vessels, it lines your digestive system, it lines your nasal cavity, it literally lines everything, okay? And so it covers organs, viscera, blood vessels, and it also makes up your secretory cells, your glands. So like your sweat glands are epithelium. Okay, so it's a lining. So you have lining in your eyes, in your nasal cavity. Your skin is epithelium. That's a lining of your entire body. The lining of your brain, the lining of organs. That is all epithelium. Okay. The next one is going to be connective tissue. Again, this is everywhere. So connective tissue is like glue. Okay. It connects things, and so that's pretty straightforward. So that's what, make sure that your skin doesn't just fall off or whatever. It's constantly connecting things, gluing things together, making sure that you are tough and structurally sound. Okay, and so connective tissue also has some specialization, so that's going to include blood, cartilage, and bone. So those are types of connective tissue. They're subtypes within connective tissue. Okay, so blood is not its own type of tissue. That's a hint. Some of y'all may see that on your quiz. Blood is not its own type of tissue. It is a subtype within connective tissue. Okay, so you have connective tissue everywhere. So you have it, in the bone in your skull. You have it in your nose. You have it under your skin, connecting everything down. Your teeth, more bone. It's also in your fat, so I guess that's what that is. That's a bunch of fat. So your fat connects everything too. So that's going to be your connective tissue. Okay. The next one is muscle. So muscle, that's what's going to generate contractile force, right? And so when everybody thinks of muscle, they think of the big muscles in your body that you can voluntarily move, right? So my biceps, my triceps, my heart, you can't voluntarily move, but it's a big chunk of muscle, right? So this is cardiac muscle. You have muscle everywhere. These are all the muscles you can voluntarily move, but you actually even have muscles, in your eyes to move your eyes around. You have muscles around your blood vessels. Okay, so that's not something you can voluntarily contract, but you do, and so that will contract and dilate your blood vessels. That's something a lot of people don't think about. You have smooth muscle in your digestive system, right? That's what's moving all the food and ingesta through your body. So there's muscle pretty much everywhere throughout the body as well. Okay. And then the last one is going to be nerve. So nerve is the one that's going to help communicate everything throughout your body. So it's specialized for transmission, reception, and integration of electrical impulses, right? So you've got nerve everywhere, obviously your brain. You've got nerve in the back of your eye, so your eyes can tell your brain what you see. You've got it in your spinal cord. You've got it all throughout your skin, all throughout your internal organs and everything. And that's so that you can feel things in your skin, and your organs can sense things, right? They can know what's going on and relay that to the rest of the body. Okay. Another thing, so this is just kind of a review on biology, right? So this is just going over, the basics, the very basics of organelles in a cell. So do just make sure that you know this, the cell membrane is the big gatekeeper. The nucleus is kind of like the brain, the computer that tells everything what to do. The mitochondrion, that's the power plant, so that's where you're making ATP, that's where you're getting energy. the endoplasmic reticulum, that's the construction team, that's building proteins and making all of these different products. And the Golgi apparatus is the packaging department. So that's what's sealing up all the products that you're making, sending them wherever they need to go. Okay, so just make sure you go over that, you understand that, and are able to match these. And that's it. So now we're just going to do some practice questions just to kind of help you guys work through these questions. And so I know this is the first quiz week and everything, so I will just kind of go over the structure of Dr. Johnson's questions, and I will tell you it will always be like this. So the way that Dr. Johnson really likes to make his questions is he likes to make them difficult. And so how it is always going to work is you're going to have 3 answer choices that are actually answer choices and things you have to think about, but it is always going to be to where you can either pick one of these, you can pick D, which is always going to say A and B, and E is always going to say A, B, and C, right? So what you're basically determining is it one of these, is it the first 2, or is it all of them? And so that is kind of the thing that makes these questions difficult, and why you have to know multiple things. So you can't just say, oh, I know this one's wrong, just kind of work through there, because if you know this one's wrong, it could still be A, it could still be B, and it could still be D. So it does make things a little difficult. So do just try to get used to how he asks his questions. There are all those questions in the question bank, so I encourage you, go over that. If you have any questions, bring them to help desk, ask your TA, and they can definitely help you work those out. Okay, so we'll do the first practice question. Which structure matches the definition? So A, the smallest unit of protoplasm. And remember, we said the smallest unit of protoplasm that can maintain life by itself is going to be a cell. So that is what this one is pointing to, right? It even says cell. So A is going to be correct, right? So a cell is the smallest unit of protoplasm. So that one's correct. We'll move on to B. Groups of cells with the same general function and texture. And it's pointing to tissue, right? And so that is correct. Remember, we have protoplasm, which makes a cell, and multiple cells with the same function and texture will make a tissue, okay? And then C, 2 or more types of tissue in a larger working unit, and it's pointing to an organ. And that one is correct too, right? Because remember, we said we get multiple different types of tissue together, and that's going to make an organ. Okay, so the answer for this one, if I can get it to work, is going to be E. So it is going to be all the above. So all of these structures match their definitions in this thing. Okay, so the next one, so these are, sorry, my cat is driving me crazy. Okay, so which micrographs illustrate carbon replica transmission electron microscopy? So yes, all of these are electron microscopy, and so now we're looking for the one that is carbon replica. So I will go ahead and tell you, so carbon replica is the one that looks like a Play-Doh mold, right? And so that one is going to be A, right? That's that lymphocyte that we're looking at. So it does look like a Play-Doh mold. It's a little carbon replica of what we've made. B and C are actually going to be scanning electron microscopy, right? So that's the one that looks like you literally scanned in an image, you took a picture or something. So the only one that is going to match is going to be A. Okay, this next one, which of the following statements about this is correct? So right here, I don't need you to know what type of cell this is and everything that's going on, but you do need to know what kind of stain this is, right? So first of all, you should be able to tell me that this is light microscopy, right? We're doing light microscopy, and then the type of stain, because it is pink and purple and everything like that, this is going to be an H&E stain, Okay, so A, this is a periodic acid shift stain. That one we can say right off the bat is false, because remember, that's the one that stains carbohydrates. That's the one that's magenta and really dark purple. It says, in this stain, nucleic acids will stain more blue. And so that is true. Remember, we said that it is that hematoxylin that stains nucleic acid, so that's why we can see all of these different nuclei. That's why they look very purpley, because they're picking up a lot of that blue stain. Okay, and then C, it says, so another little trick, so if I said this one is wrong, A is wrong and B is right, that means that C better be wrong, because there is no option where only B and C are right? So if I know that B is right, A is wrong, C better be wrong. So that's how I can kind of check myself. So we're hoping that C isn't going to be right. Let's see. It says, in this stain, proteins will stain more blue. Yeah, so that's going to be false, right? We said it's nucleic acids that are going to stain more blue. Proteins are going to stain more red, right? That's that eosin in the H&E stain, where these proteins, these areas that are very rich in proteins, are going to stain more red. So the answer for this one is just going to be B. Okay, this one, which item description about magnification is correct? So it says magnification is an increase in image size. And remember, that is what we said about magnification, is it is just making things bigger. So that is correct. Resolution, it's saying, is the largest distance between 2 points that can be distinguished. And remember, that is not what we said about resolution. It's this one word right here that's changed. Resolution is the smallest distance between 2 points that can be distinguished, right? It's really easy to tell the difference between points that are really far away. We want to tell the difference between parts that are really close together. So it's the smallest distance. That's going to be our resolution, okay? And then this one right here, it says magnification, smallest distance between 2 points that can be distinguished. So he's trying to be tricky here, right? So this is actually the correct definition for resolution, the smallest distance between 2 points that can be distinguished, but not for magnification, right? So both of those are incorrect. So the answer is just A. Okay. As always, you can read this. Here are all of the resources that are available to you and everything. There's no way I can cover everything that's going to be on your quiz this week, but I am trying to do my best to summarize everything, make things easy for you guys. If you do have any questions, feel free to reach out. Don't be overwhelmed. This first week, I will tell you, is going to be a little bit easier. The questions are a little more straightforward, and that is because, we're trying to introduce you to this course, get you comfortable with microscopy and everything before we actually start teaching you things about the cells that you're looking at. Okay? So hopefully this helped you guys cover things. I know a lot of it was pretty straightforward, but it is the next week that we are going to kind of get a little more in depth and hit the ground running. Okay, so good luck with your quiz, good luck with this week, and thanks for watching.